Advanced Neurology. So this lecture goes through reflexes and then the autonomic nervous system, and then it ends with talking a little bit about senses. So reflexes are quick, involuntary, stereotyped reactions of glands or muscles to stimulation. So quick, they're instantaneous. They're involuntary, so they happen without us thinking about them. And they're stereotyped, which means they're predictable. Time after time, they're going to do the same thing. Reflexes require stimulation, not spontaneous actions, but responses to sensory input. Reflexes are quick. They involve few, if any, interneurons and minimum synaptic delay. Synaptic delay is talking about minimum time to travel, or for the signal to travel through a synapse. And the synapse, if you remember, is the space between adjacent neurons. They're involuntary, occur without intent, and are difficult to suppress. And they're stereotyped, occur essentially the same way every time. Reflexes include glandular secretion and contraction of all three types of muscle. Somatic reflexes are those reflexes involving the somatic nervous system, innervating skeletal muscles, while visceral reflexes are reflexes involving organs such as the heart and intestines. So somatic reflexes typically cause the movement of something that's normally under voluntary control. So even though the reflex itself is not voluntary, it's an automatic thing that happens, normally the muscles that are stimulated through a somatic reflex would be under voluntary control. So if I prick my finger on something sharp, I'm going to immediately pull it away as a reflex. Now normally those muscles to move my arm would be under voluntary control, but they take over in the somatic reflex process that becomes an automatic process that happens. So somatic reflexes typically would cause a movement of something that we would normally be able to control voluntarily, while visceral reflexes, that causes the movement of something inside the body that we would not normally be able to control. Somatic reflex involves a reflex arc with the following pathway. So there's receptors in the skin, muscles, or tendons. An afferent nerve fiber carries information from the receptors to the posterior horn of the spinal cord, or the brainstem. There's an integrating center, a point of synaptic contact between neurons in the gray matter of the cord or brainstem. And that determines whether efferent neurons issue signals to muscles. Efferent nerve fibers then carry the motor impulses to the muscles, and effectors are the muscles that carry out the response. So this is that pathway. So here you can see the stimulus would be stretching this receptor in the muscle. And when that stretch receptor picks up that signal, then we're going to have an afferent or sensory nerve fiber take that information to the back of the spinal cord. Here it's going to enter into an interneuron that is inside of the central nervous system, and it's synapsing in that integration center, that interneuron happening here. Then a response is going to be determined, and if the response is necessary, then we will have this efferent or motor nerve fiber stimulate the contraction of this muscle to move it away from the source. Contracting this muscle causes the leg to kick out, so that would be the response. The effector is the muscle itself. The muscle spindle. So many somatic reflexes involve muscle spindles. They're stretch receptors that are embedded in skeletal muscles. They serve as proprioceptors, specialized sense organs to monitor position and movement of body parts. They inform the brain of muscle length and body movement, enable the brain to send motor commands back to the muscles that control coordinated movement, corrective reflexes, muscle tone, and posture. So this shows a picture of a muscle spindle and its innervation. You can pause the video and take a look at all the different components. Intrafusal fibers are modified muscle fibers within the spindle. The rest of the muscle's fibers, those generating force or movement, are extrafusal fibers. A gamma motor neuron innervates the ends of an intrafusal fiber and keeps it taut. Alpha motor neurons supply the extrafusal fibers. The mid portion of the intrafusal fiber contains the sensory nerve fibers. There's primary afferent fibers, which monitor fiber length and the speed of the length changes. Secondary afferent fibers monitor length only. Example of a spindle function, they help you keep upright when standing on a boat. The stretch reflex, so stretch or myotactic reflex. When a muscle is stretched, it fights back and contracts, helps maintain equilibrium and posture. The head starts to tip forward as you fall asleep, muscles contract to raise the head. Stabilize joints by balancing tension in extensors and flexors, smoothing muscle actions. Stretch reflex is mediated primarily by the brain, but its spinal component can be more pronounced if muscle is suddenly stretched by a tendon tap, knee jerk. Monosynaptic reflex arcs, one synapse between the afferent and efferent neurons. Little synaptic delay, so a very prompt response is produced. Patellar or knee jerk reflex is a monosynaptic reflex. Mono meaning one, synaptic is talking a synapse, so that's where we get the one synapse from. Testing somatic reflexes helps diagnose many diseases. Reciprocal inhibition, reflex phenomenon that prevents muscles from working against each other by inhibiting antagonist when agonist is excited. This shows a diagram of the patellar reflex. So you tap on the patellar ligament that excites nerve endings in the muscle spindle in the quadriceps. Stretch signal travels to the spinal cord by the afferent fiber and the dorsal root. Primarily, afferent neuron stimulates alpha motor neuron in the spinal cord. Efferent signals in the alpha motor neuron or motor nerve fiber stimulate quadriceps to contract, producing a knee jerk. At the same time, a branch of the afferent nerve fiber stimulates inhibitory muscle neuron or motor neuron in the spinal cord. That neuron inhibits alpha motor neuron that supplies hamstring muscles, and hamstring contraction is inhibited, so hamstrings or knee flexors do not antagonize the quadriceps. The flexor or withdrawal reflex. Flexor reflex is the quick contraction of flexor muscles resulting in the withdrawal of a limb from an injurious stimulus. Triggers contraction of the flexors and relaxation of the extensors in that limb. Postsynaptic reflex arc, pathway in which signals travel over many synapses on their way to the muscle. Crossed extension reflex, contraction of extensor muscles in limb opposite of the one that is withdrawn. It maintains balance by extending the other leg. Flexor reflex uses an ipsilateral reflex arc, the stimulus and response on the same side, whereas crossed extension reflex uses a contralateral reflex arc. Input and output are on opposite sides. Intersegmental reflex, one in which the input and output occur at different levels, segments of the spinal cord. Pain in the foot causes contraction of abdominal muscles. So here you can see a diagram showing the flexor and crossed extension reflexes. You can go ahead and pause the video and take a look at this diagram. The tendon reflex. So tendon organs, proprioceptors in a tendon near its junction with a muscle involved in the tendon reflex. It's 0.5 millimeters long, consists of encapsulated bundle of collagen fibers and one or more nerve fibers. The tendon reflex responds to excessive tension on the tendon, inhibits muscle from contracting strongly, moderates muscle contraction before it tears a tendon or pulls it loose from the muscle or bone. So that's just a kind of brief overview of some of our reflexes. So if we damage or injure the spinal cord, we will have issues with reflexes. A complete transection, complete severance of the cord. You have immediate loss of motor control below the level of injury. Above C4 poses the threat of respiratory failure and spinal shock. Paralysis comes in three forms: paraplegia, paralysis of both of the lower limbs; quadriplegia, paralysis of all four limbs; and hemiplegia, paralysis on one side of the body. Paresis is partial paralysis or weakness of the limbs. The autonomic nervous system. So the autonomic nervous system, ANS, is a visceral motor system, a motor nervous system that controls glands, cardiac muscle, and smooth muscle. The primary target organs of the ANS include the viscera of the thoracic and abdominal cavities, some structures of the body wall, cutaneous blood vessels, sweat glands, and erector muscles. It carries out actions involuntarily without our conscious intent or awareness. Visceral effectors do not depend on the ANS to function, but only to adjust their activity to the body's needs. Severing ANS nerves to organs results in exaggerated responses, denervation, hypersensitivity. Visceral reflexes. These are unconscious, automatic, stereotyped responses to stimulation involving visceral receptors and effectors controlled by ANS. The visceral reflex arc is as follows. So the receptors are nerve endings that detect stretch, tissue damage, blood, chemicals, body temperature, and other internal stimuli. Afferent neurons lead to the CNS. The integrating center are the interneurons in the CNS. Efferent neurons carry motor signals away from the CNS, and effectors carry out end response. This is a diagram of an autonomic reflex arc. So as an example, baroreceptors sense increased blood pressure. So baroreceptors are receptors that sense pressure. That sends a signal down the glossopharyngeal nerve to the medulla oblongata. The medulla oblongata contains an integrating center with interneurons. And then the vagus nerve transmits inhibitory signals to the cardiac pacemaker. The effector, the heart rate, is going to decrease. The ANS has two subsystems, sympathetic and parasympathetic. The sympathetic division prepares the body for physical activity, exercise, trauma, arousal, competition, anger, or fear, also known as the fight or flight reaction. It increases heart rate, blood pressure, airflow, blood glucose levels, etc., reduces blood flow to the skin and the digestive tract. If you are experiencing a need to fight or run away from something, your digestive system is going to shut off. Usually you only digest food when you're at complete rest. Blood instead is going to be routed to your muscles because your muscles are going to be needed to move or to fight. The skin. So we don't want to have a lot of blood near the surface of our body because if we are fighting, we don't want to get an injury and then bleed out from that injury. So blood is routed away from the skin to our effector muscles. The parasympathetic division calms many body functions, reducing energy expenditure and assists in daily or bodily maintenance, resting and digesting state. This is when we have digestion and waste elimination happen. So here you can see the difference between the somatic and autonomic, the pathway and the effectors involved. So somatic, we would have thick, heavily myelinated fibers. In autonomic, we would have preganglionic in the central nervous system, which is thin and lightly myelinated, and postganglionic, unmyelinated axon, which extends to the effector. In the somatic nervous system, the effectors would be skeletal muscle primarily, and in the autonomic, we're talking about involuntary muscles such as smooth and cardiac, glands, visceral organs, etc. Of the autonomic nervous system, we have the parasympathetic and sympathetic divisions. So here you can see a major, all the major differences listed. So the role, parasympathetic would be rest and digest. Sometimes we refer to these as the 3 Ds: digestion, defecation, and diuresis. Diuresis is fluid balance. Sympathetic, fight or flight, the 4 E's: exercise, excitement, emergency, or embarrassment. Types of effects: parasympathetic are short-lived, highly localized control. Sympathetic, long-lasting, body-wide effects. Parasympathetic would cause the constriction of pupils and close vision. Sympathetic dilates pupils and aids in far vision. So why would we dilate the pupils? So we can see more. Why would we want to see far? So we can look for escape routes. So sympathetic is all about fight or flight. Under normal conditions, normal resting conditions though, we don't want our pupils dilated because any amount of light that comes in there can damage the retina. So we would constrict the pupils, and then we only really need to see close up under normal circumstances. Glands. Parasympathetics, it's going to stimulate secretion. Sympathetic, it inhibits secretion. It's going to move blood away from these areas. Why? Because we're not going to need glands. All we're going to need to do is beef up our blood supply to our muscles so that we can have enough oxygen flowing to them to produce energy to be able to run or fight. Sweat glands, parasympathetic has no impact on, but sympathetic would stimulate. Adrenal medulla, again, parasympathetic none. Sympathetic would increase the secretion of epinephrine and norepinephrine. The erector pili, which are those tiny little muscles that cause your hairs to stand up on end, no effect for the parasympathetic, but they would cause goosebumps or the hairs to stand up in the sympathetic. Now, why would they cause that to happen? If we think back in an evolutionary time frame, and if we think about some of our animal close relatives, hair standing up on end makes us look scary and bigger. So even though someone getting goosebumps isn't a scary thing today, it used to be when we used to be super hairy. So that kind of makes us look bigger and scarier. The heart, parasympathetic would decrease the heart rate while sympathetic would increase the heart rate. Why do we want a fast heart rate? Again, the whole goal is to circulate oxygen around fast enough to replenish the oxygen supply in our muscles so that our muscles can make energy. Bladder, parasympathetic promotes voiding or urinating. Sympathetic inhibits that. In the lungs, parasympathetic constricts the bronchioles. We don't want our bronchioles wide open. Why? Because the wider open our bronchioles are, the higher incidence of pathogens getting in. So under normal resting conditions, we want those things kind of constricted, but sympathetic responses dilates bronchioles, so we want more oxygen coming into the body. At this point, we care about staying alive and not about fighting infection. The GI tract, parasympathetic is stimulatory, sympathetic is inhibitory. So by now, you should know, like rest and digest. Digest has to do with the GI tract. Liver, glucose uptake for parasympathetic, glucose release for sympathetic. We're releasing glucose from the liver so that we can fuel our fight or flight response. The gallbladder, parasympathetic, stimulatory, sympathetic, inhibitory. The kidney, parasympathetic has no impact, sympathetic has renin release to decrease urine output. The penis, erection would be under parasympathetic. Sympathetic is responsible for ejaculation. Blood vessels, no impact from the parasympathetic side, but on the sympathetic side, constriction to increase blood pressure and push blood towards skeletal muscles and away from the glands and skin. Autonomic tone, normal background rate of activity that represents the balance of the two systems according to the body's needs. The body does not simply alternate between states, action versus rest. Examples of parasympathetic tone: maintain smooth muscle tone in the intestines, hold the resting heart rate down to about 70 to 80 beats per minute. Example of sympathetic tone: keeps most blood vessels partially constricted and maintains blood pressure. Neither division has universally excitatory nor inhibitory effects. The ANS has components in both the central and peripheral nervous systems, control nuclei in the hypothalamus and other brainstem regions, motor neurons in the spinal cord and peripheral ganglia, nerve fibers that travel through the cranial and spinal nerves. The ANS motor pathway versus somatic motor pathway. In somatic pathway, a motor neuron from the brainstem or spinal cord issues a myelinated axon that reaches all the way to the skeletal muscle. In the autonomic pathway, a signal must travel across two neurons to get to the target organ. and cross a synapse where these two neurons meet in an autonomic ganglion. The first neuron in the pathway, the cell body in the brainstem or spinal cord, its axon, preganglionic fiber, extends to the autonomic ganglion. At the ganglion, the first neuron synapse with the second neuron. Acetylcholine is the neurotransmitter. The second neuron in the pathway, cell body in autonomic ganglion, its axon, postganglionic fiber, extends to the target cells. Neurotransmitter is either acetylcholine or norepinephrine. So this shows a somatic motor innervation, and you can see acetylcholine. Well, here you can see the comparison of the sympathetic and parasympathetic motor innervation. You can pause the video and kind of click back and forth between these two different slides. The sympathetic division. So the anatomy of the sympathetic division, its pathways arise from the thoracic and lumbar regions of the spinal cord. Relatively short preganglionic and long postganglionic fibers. The general pathway, cell bodies of preganglionic neurons in lateral horns and nearby regions of the spinal cord, gray matter. Fibers exit the spinal cord by way of spinal nerves T1 through L2. They lead to near-back sympathetic chain of ganglia, the paravertebral ganglia, a series of longitudinal ganglia adjacent to both sides of the vertebral column from the cervical to coccygeal levels. Usually, 3 cervical, 11 thoracic, 4 lumbar, 4 sacral ganglia, and 1 coccygeal ganglion. Sympathetic nerve fibers are distributed to every level of the body. And here's a diagram that depicts that whole summary, so you can kind of pause the diagram and take a look at this. Anatomy of the sympathetic division continued. So each paravertebral ganglion is connected to a spinal nerve by 2 branches, the communicating rami. Preganglionic fibers are small myelinated fibers that travel from the spinal cord to the ganglion by way of white communicating ramus. Postganglionic fibers leave the ganglion by way of the gray communicating ramus and by other routes. The gray ramus returns to the spinal nerve. The postganglionic fibers extend the rest of the way to the target organ. And here's a diagram that you can pause the video and take a look at these different parts. Sometimes looking at diagrams and looking at the pictures makes things click a little bit more than just reading a list of definitions. So definitely pause the video, take some time to review the different pathways here. Nerve fibers leave the sympathetic chain by 3 routes: spinal, sympathetic, and splanchnic nerves. The spinal nerve route, some postganglionic fibers exit a ganglion by way of the gray ramus, return to the spinal nerve, and travel the rest of the way to the target organ. Most sweat glands, erector muscles, and blood vessels of the skin and skeletal muscles. The sympathetic nerve route, some postganglionic fibers leave by sympathetic fiber nerves that extend to the heart, lungs, esophagus, and thoracic blood vessels. These nerves form carotid plexus around each carotid artery of the neck. Issue fibers from there to the effectors in the head, sweat, salivary, nasal, erector muscles, blood vessels, dilators of the iris. Some fibers of the superior and middle cervical ganglion form cardiac nerves to the heart. The splanchnic nerve route, some fibers arise from the spinal nerves T5 to T12, pass through the sympathetic ganglion without synapsing. They continue on as the splanchnic nerves, lead to the second set of ganglia, the collateral ganglia, and synapse there. Again, you can take a look at the diagram. Collateral ganglia contribute to a network called the abdominal aortic plexus. It wraps around the abdominal aorta, and there's 3 major collateral ganglion in this plexus: the celiac, superior mesenteric, and inferior mesenteric ganglia, located at points where arteries with the same names branch off of the aorta. Postganglionic fibers accompany these arteries and the branches to the target organs. The solar plexus, collective name for the celiac and superior mesenteric ganglia. Nerves radiate from the ganglion like rays of the sun. Here you can take a look at that. And here's the abdominal aorta. that's going to run through the back of the abdominal cavity. Neuronal divergence predominates in the sympathetic division. Most sympathetic preganglionic neurons synapse with 10 to 20 postganglionic neurons. One preganglionic neuron can excite multiple postganglionic fibers, leading to different organs, target organs. It has relatively widely spread effects. The adrenal glands, so adrenal or suprarenal glands. Suprarenal is talking about on top of the renal. Adrenal, renal, again, you see that word. Like I keep mentioning, anytime you break down this part, the big terms into their stems, you can kind of start to deduce definitions. So adrenal, paired glands located on the superior poles of the kidneys. Each is 2 glands with different functions. There's an adrenal cortex on the outside and adrenal medulla on the inside. The cortex, the outer layer, secretes steroid hormones, while the medulla, the inner core, essentially is a sympathetic ganglion. It consists of modified postganglionic neurons without dendrites or axons, stimulated by preganglionic sympathetic neurons. It secretes a mixture of hormones into the bloodstream. The catecholamines, about 85% epinephrine, also known as adrenaline, and 15% norepinephrine, also known as noradrenaline. Sympathoadrenal system, collective name for the adrenal medulla and sympathetic nervous system, closely related in development and function. Anatomy of the parasympathetic division. Its pathways arise from the brain and sacral regions of the spinal cord. Relatively long preganglionic and short postganglionic fibers. The general pathway is listed here. So the cell bodies of preganglionic neurons in the midbrain, pons, and medulla or sacral spinal cord segments S2 to S4. Preganglionic fibers travel through the cranial or sacral nerves and in the trigeminal ganglia in or near target organs. The synapse with the postganglionic neurons in the ganglia and postganglionic fibers cover the rest of the short distance to the target organ. Parasympathetic division is relatively selective in stimulation of the target organ. Very little neural divergence. Parasympathetic fibers leave the brainstem in the following cranial nerves: oculomotor, facial, glossopharyngeal, and vagus. Remaining parasympathetic fibers arise from S2 to S4 of the spinal cord. Fibers travel in anterior rami of spinal nerves, then form pelvic splanchnic nerves that lead to inferior hypogastric plexus. Some fibers synapse at this plexus. Most travel through the pelvic nerves to the terminal ganglia. Target organs would be distal half of the colon, rectum, urinary bladder, and reproductive organs. And here's a diagram that you can pause the video and review. The enteric plexus, also known as the enteric nervous system, the nervous network of the digestive tract, composed of 500 million neurons found in the walls of the digestive tract. Unlike the rest of the ANS, it does not arise from the brainstem or spinal cord. Like the rest of the ANS, it innervates smooth muscle and glands, but has its own ganglia and reflex arcs, regulates motility of the esophagus, stomach, and intestines, and secretion of digestive enzymes and acid. Normal digestive function also requires regulation by sympathetic and parasympathetic systems. Megacolon. Hirschsprung disease is a hereditary defect causing the absence of the enteric plexus. There's no innervation in the sigmoid colon and rectum, constricts permanently, and will not allow passage of feces. Feces becomes impacted above constriction. Megacolon is a massive dilation of the bowel accompanied by abdominal distension and chronic constipation. May be colonic gangrene, perforation of the bowel, and infection of the peritoneum, usually evident in newborns who fail to have their first bowel movement. All right, autonomic effects on target organs. Divisions of the ANS have opposite effects on an organ. 2 reasons this is possible. Sympathetic and parasympathetic fibers secrete different neurotransmitters. The receptors on target cells vary. Target cells respond to the same neurotransmitter differently depending on what type of receptor they have for it. There are two different classes of receptors for acetylcholine and two classes of receptors for norepinephrine. Acetylcholine is secreted by all preganglionic neurons in both divisions and by postganglionic parasympathetic neurons. Neurons that secrete acetylcholine are called cholinergic neurons. Any receptor that binds acetylcholine is called a cholinergic receptor. There's two categories of cholinergic receptors. Muscarinic receptors found in cardiac muscle, smooth muscle, and gland cells with cholinergic innervation. They act through second messenger systems. Receptor subtypes exist, often providing contrasting effects on organs. Example, acetylcholine excites intestinal smooth muscle but inhibits cardiac muscle. Nicotinic receptors are found on postganglionic neuron cell bodies in autonomic ganglia on the cells of adrenal medulla and neuromuscular junction of skeletal muscle fibers. Binding of acetylcholine to the receptor is always excitatory. Open ligand-gated ion channels produce excitatory postsynaptic potentials. Norepinephrine is secreted by nearly all sympathetic postganglionic neurons. Nerve fibers that secrete norepinephrine are called adrenergic fibers. Receptors for norepinephrine are called adrenergic receptors. Alpha adrenergic receptors, usually excitatory. There's two subclasses which use different second messengers, and then beta adrenergic receptors, usually inhibitory. Two subclasses with different effects, but both act through cyclic AMP as a second messenger. Alpha adrenergic receptors are usually excitatory, beta usually inhibitory. Autonomic effects on glandular secretion are often an indirect result of their effect on blood vessels. Increased blood flow, increased secretion, decreased blood flow, decreased secretion. Sympathetic effects tend to last longer than parasympathetic effects. Norepinephrine released by sympathetic fibers can be reabsorbed by the axon terminal, diffuse to adjacent tissues, or pass into the bloodstream, whereas acetylcholine from parasympathetic fibers is broken down quickly at the synapse. Some ANS fibers release other neurotransmitters in addition to acetylcholine or norepinephrine. Examples are listed here. And here's a diagram that you can kind of see the differences depicted. Dual innervation. So most viscera receive nerve fibers from both parasympathetic and sympathetic divisions. In that table that I went over, you see that some of these do have innervation from both, while one is specifically for parasympathetic or one is specifically for sympathetic. They have antagonistic effects. They oppose each other. They can be exerted through dual innervation of the same effector cells. They can be exerted because each division innervates different cells. Cooperative effects means two divisions act on different effectors to produce a unified overall effect. Example would be saliva production. Even when both divisions innervate a single organ, innervation is not always equal or exert equal influence. And here you can see a diagram of the iris and the dual innervation here. Dual innervation not always necessary for the ANS to produce opposite effects. Some effectors receive only sympathetic fibers. Example, regulation of blood pressure and routes of blood flow. Baseline sympathetic tone maintains vasomotor tone, vessels in a continual state of partial constriction. Increasing firing frequency causes vasoconstriction. Decrease in firing frequency causes vasodilation. Can also shift blood flow from one organ to another as needed. During stress, blood vessels to muscles and heart dilate, while blood vessels to the skin constrict. And here's a diagram that you can pause the video and review. Central control of autonomic function. The cerebral cortex and limbic system, emotions, influence the ANS because of connections between our limbic system and hypothalamus. The hypothalamus contains nuclei for primitive functions like hunger, thirst, sex. Midbrain pons and medulla oblongata contain nuclei for cardiac and vasomotor control, salivation, swallowing, sweating, bladder control, and pupillary changes. Spinal cord, reflexes for defecation, and micturition. We control these functions because of our control over skeletal muscle sphincters. If the spinal cord is damaged, the smooth muscle of the bowel and bladder is controlled by autonomic reflexes built into the spinal cord. Drugs in the nervous system. So neuropharmacology is the study of the effects of drugs on the nervous system. Sympathomimetics enhances sympathetic activity. They stimulate receptors or increase norepinephrine release. Cold medicines that dilate the bronchioles or constrict the nasal blood vessels. Sympatholytics suppress sympathetic activity. Block receptors or inhibit norepinephrine release. Beta blockers reduce high blood pressure, interfering with effects of epinephrine and norepinephrine on the heart and blood vessels. Sympathomimetics enhance parasympathetic effects, while parasympatholytics suppress them. Many drugs also act on neurotransmitters or their receptors in the CNS. Prozac is a selective serotonin reuptake inhibitor, SSRI, which blocks the reuptake of serotonin to prolong its mood-elevating effect. MAO inhibitors block the enzyme monoamine oxidase to slow the breakdown of monoamine neurotransmitters, which aids in treating depression. Caffeine competes with adenosine, in the presence of which causes sleepiness by binding to its receptors. So with caffeine, it competes with adenosine. Adenosine makes you sleepy. Caffeine prevents it competes with it so that you don't get sleepy. All right, so then moving on to our last section, sensory receptors. So receptor are the structures specialized to detect a stimulus. Some receptors are bare nerve endings. Others are true sense organs, the structure that combines nerve tissue surrounded by other tissues that enhance response to certain type of stimulus. Accessory tissues may include added epithelium, muscular, or connective tissue. They range in size and complexity, eye and ear versus dendrite wrapped in a little bit of connective tissue. Transduction, the conversion of one form of energy to another. This is the fundamental purpose of any sensory receptor. They convert stimulus energy, such as light, heat, touch, sound, into a nerve signal. There's two stages in the sensory process. One is sensation, and one is perception. Sensation is when a sensory receptor detects the stimulus, which creates a small local electrical charge, or change, the receptor potential. If the receptor potential is strong enough, the neuron fires action potentials and nerve signals to the brain. Perception is the conscious experience and interpretation of a stimulus. Not all sensations lead to perception. Many signals are filtered out before reaching the cortex. Sensory receptors transmit four kinds of information: modality, location, intensity, and duration. Modality is the type of stimulus or the perception it produces. Examples include vision, hearing, taste. It's determined by which region of the brain is wired to receive the information. Location is where a stimulus is located, is encoded by which nerve fibers are firing. The receptive field, an area within which a sensory neuron detects stimuli. Size of a receptive field determines resolution, the ability to distinguish between two close together stimuli. The neurons in the fingertips have small receptive fields, allowing for fine two-point touch discrimination. So this is that two-point touch discrimination test. Information transmitted by receptors. So intensity, strength of the stimulus. Examples: whether a sound is loud or soft, a light is bright or dim, a pain is mild or excruciating. It's encoded in three ways: which fibers respond, how many fibers respond, and how fast the fibers are firing. Duration, how long the stimulus lasts, encoded by changes in firing frequency over time. There is such a thing called sensory adaptation. So if a stimulus is prolonged, firing of the neuron gets slower over time and we become less aware of it. We adapt to it. Phasic receptors adapt quickly, burst of signals when stimulus starts, and then reduce or stop signaling even if the stimulus continues. Tonic receptors adapt slowly. Action potentials continue more steadily while the stimulus is present. Classification of receptors. So there's thermal receptors, which respond to heat and cold. Photoreceptors respond to light. These are only found in the eyes of people. Nociceptors respond to tissue injury, situations threatening damage. They activate processes, or activation produces sensation of pain. Chemoreceptors respond to chemicals, and chemicals can be in the form of odors, tastes, or body fluid composition. Mechanoreceptors respond to physical deformation. This is vibration, touch, pressure, stress, tension, includes organs of hearing and balance. We can classify by the origin of stimuli. Exteroceptors sense stimuli external to the body. Interoceptors detect internal stimuli. And proprioceptors sense body position and movements. General somatosensory or somaesthetic senses are widely distributed in the skin, muscles, tendons, joints, and viscera. Special senses are limited to the head, and they're innervated by cranial nerves and involve complex sense organs. General senses. So unencapsulated nerve endings are dendrites with no connective tissue wrapping. There's free nerve endings, bare dendrites that detect temperature and pain. Tactile or Merkel discs, flattened nerve endings that terminate at tactile cells in the basal layer of the epidermis. Hair receptors, root hair plexuses, dendrites coiled around a hair follicle and respond to movements of hair. Then there's encapsulated nerve endings, nerve fibers wrapped in glial cells or connective tissue. Wrapping enhances sensitivity or selectivity of the response. Tactile Messner corpuscles, 2 or 3 nerve fibers within the fluid-filled capsule of flattened Schwann cells. End bulbs, Krause end bulbs, sensory nerve fibers surrounded by connective tissue sheath. Bulbous or Ruffini corpuscles, flattened elongated capsules containing a few myelinated axons in incomplete fibrous capsule. Lamellar or Pacinian corpuscles, large ovoid receptors with a single dendrite surrounded by layers of flattened Schwann cells and fibroblasts. This is a really good diagram that depicts all of the different types that I just defined. You can pause the video and take a look at these. Sensory projection, so transmission of information from the receptor to the specific location in the cerebral cortex. Signals travel along a projection pathway. Most somatosensory signals travel by way of 3 neurons. A first-order neuron, signals from the head travel to the pons or medulla via cranial nerves. Signals from below the head enters the posterior horn of the spinal cord via spinal nerves. Touch, pressure, and proprioception fibers are large, myelinated, and fast. Heat and cold fibers are small, unmyelinated, and slower. Second-order neuron decusate to the opposite side in the spinal cord, medulla, or pons. They end in the thalamus, except for proprioception, which ends in the cerebellum. Third-order neuron, thalamus to the primary somaesthetic cortex of the cerebrum. Pain is the unpleasant perception of actual or potential tissue damage. Typically leads to evasive action, protection, loss. It's lost in leprosy and diabetes mellitus. Nerve damage can result in the neglect of injuries. Types of pain. There's nociceptive pain and neuropathic pain. Nociceptive pain stems from tissue injury, occurs when nociceptors are activated, while neuropathic pain stems from injuries to nerves, spinal cord, meninges, or brain. Nociceptive pain subdivided into 3 kinds by origin. Visceral pain arises from internal organs. It's diffuse, dull, and hard to locate. That's why, like when kids get tummy aches, they just say, my tummy hurts, and it's really hard for them to say exactly where it hurts. It's diffuse pain. Deep somatic pains arises from bones, joints, muscles. Superficial somatic pain usually arises from the skin. Two types of nerve fibers involved in pain responses. Fast pain, immediate, sharp, localized pain. Slow pain is burning, dull, and aching pain. The projection pathways for pain. Pain signals from the head travel to the brainstem via cranial nerves. First-order neurons, second-order neurons, and third-order neurons are going to travel in a different path. Pain signals from the neck and below travel through three ascending tracks: spinothalamic, spinal reticular, or gracile fasciculus. And here is a diagram of pathways of pain. And then you can see where first order, second order, and third order nerve fibers are found. Referred pain is pain in the viscera that's often perceived as originating from a superficial site, such as the skin. Results from convergence of neural pathways in the central nervous system. The brain cannot distinguish the source. Example, heart pain felt in the shoulder or arm because both send pain input through spinal cord segments T1 through T5. And here you can see referred pain and where it's typically felt. CNS modulation of pain. Pain is subjective, highly variable, and influenced by physical and mental state. CNS has analgesic or pain-relieving mechanisms. Endogenous opioids are analgesic peptides that are secreted by the CNS, pituitary, and digestive tract. They act as neuromodulators that block pain and give pleasure. Enkephalins, endorphins, and dynorphins are examples. Opioids block pains through spinal gating. It stops pain signals at the posterior horn of the spinal cord. Interneurons are stimulated to release enkephalins, inhibit second-order pain neurons. Two gating mechanisms. Descending analgesic fibers arise in the brainstem, descend in reticulospinal tract, and activate inhibitory spinal interneurons. Rubbing or massaging injury, mechanoreceptors in the skin and deeper tissues can activate inhibitory spinal interneurons. And then here is a diagram that you can pause and take a look at and kind of follow through all the pathways.